Recording.
OK，那是我先开始吗？我来做 introduction。
对，可以直接开始。
OK，one second，我来 figure out 怎么 share screen。OK，大家可以看到吗
可以开始。
？Great。那我们今天要讲的，我是用中文还是用英文？有没有一个
Up to
说
you, up to you。
up to me？我先介绍一下，就是今天我们讲的是 tissue engineering，然后由我来讲 background，然后是5亿来做一篇 journal，分享和Sean来做最后的 industry，分享。OK，那我就开始了。OK，我还是用英文吧。So what is tissue engineering? So it is a subfield, from my understanding, from biomaterials and regenerative medicine, and it refers to the practice of combining scaffolds, cells, and biologically active molecules into functional tissues. And this is a diagram that usually represents the flow of work. Okay, and what are some typical applications? There's quite a few because this field has been established for quite a long time. And the first three categories are more traditional, that they focus on bone and cartilage repair, cardiac and liver tissue engineering, and lastly, drug delivery. This one, from my understanding, is because of the scaffolding that's being built to facilitate the tissues that's being grown in bioreactors. And drug delivery is also a major part, also a crucial part in helping the growth factors later being distributed into the growing tissues to help them and guide them to grow. Okay, so there are some more recent and interesting developments in the field that these two are some of them that I found to be interesting. One of it is the unit programmability of tissue engineering. And second one is the minimal invasive soft tissue repair using shrunken scaffolds. And what are some what is the scaffolding and what is growth factors that I just talked about? Let's dive into that in the next slide. Okay, sorry, workflow. Let's go through the workflow first. So to suit the demands of restore, replace, or regenerate of damaged tissues or whole organs, these workflows on the right is usually adopted. So suitable biomolecules and living cells are selected, and then they're incorporated into scaffolds, which is then placed into a bioreactor to grow. And cultivation supports tissue growth prior to implanting. And lastly, scaffold is expected to biodegenerate, and ideally post-implanting into the patients. And that is the basic workflow of tissue engineering. And next slide is that we're going to talk about quite more detailedly about what is a scaffold or slash matrix and all the other components that goes into the workflow. Okay, so scaffold or matrix are adopted to mimic ECM, which most cells in our body are growing, except for blood cells, are growing in these extracellular matrices in natural tissues. And for ECMs, their purposes are to provide structural support for cells to reside, and et cetera, that are listed here. I believe people are in biology or more biomedical engineering might know them. So I'm not gonna go to the detail about it. You can take a few moments to read it if needed. And to suit these to suit these demands of ECMs, that scaffolds are supposed to provide the same functions as the extracellular matrices. So in these parts, that ECMs are usually act as reservoirs of growth factors, or scaffolds can usually deliver growth factors, and they don't have to act as reservoirs. They only have to be able to deliver these chemicals into the tissues that are growing. And lastly, ECMs are expected to have a flexible physical environment, and for our artificial scaffolds, that we can just simply set volumes for tissues to grow. Okay, the next part is cells and growth factors. And I think these are pretty straightforward, that you need cells that are capable of generation and growth factors to stimulate them to grow the way we want them to. And in this picture here, is basically showing how a growth factor can help a cell to be mobilized on the matrix. And again, growth factors are a group of peptides that can mediate cellular proliferation, migration, and differentiation by binding to these receptors on the cells. And these are just basically cell signaling. And as from background, I'm not going to dive too much into how each type of growth factors can influence growth. And because of the demands for tissue engineering are various and broad. And so the growth factors to them are also have a broad spectrum, that for each case, there's going to be different growth factors. For example, for cartilage, there are specific growth factors that are promoting cartilage growth. So for each case that we have mentioned in the application, the growth factors will be like drastically different from each other. And lastly, this topic is very near and dear to me, bioreactors. Bioreactors are used in tissue engineering in order to mimic the natural physiological environment of cells. Just like scaffolding and growth factors, all these compartments, engineering compartments or systems are here to help the tissues grow in a very artificial environment, but mimicking the natural physiological environment where the tissues are supposed to grow. So bioreactors are supposed to provide biochemical and physical regulatory signals and direct cells to differentiate and provide a suitable platform for them to develop new tissues by stimulating EC, extracellular matrix production. And once again, cells don't have to completely rely on scaffolding. They can also produce their own ECMs. Some do, some does not. Depends on the exact situations that you're in. So in this diagram, this is a part of the diagram from a Nature article that's saying what a closed bioreactor system should look like. After taking samples from the patient and analyzing our needs, we can place tissues into this kind of system where fresh media goes in and waste goes out. And there's pump and sensors to regulate the entire system. And as we are in the human system, the oxygen is also quite needed and need to be regulated in the system as well. And unlike common industrial scale bioreactors, these lab scale bioreactors are quite small, and for each different type of tissues, the needs are quite different. So people usually use 3D printers to design their own reactors as a more economic friendly and more a faster way to produce these. Yeah, that's basically the end of my slides. Please correct me if there's anything wrong about it. I'm also new to this topic. Okay.
OK，然后就到我。就这 introduction 讲的非常好，就是后面我讲的这个 journal，它基本上就是会把每一个环节给一个具体的例子，就还蛮 related 的。
好耶！
不错不错，让我来 share 一下 screen。OK， can people see me? See my screen? Okay, cool. So I'm just gonna share an article from our lab, from my lab, Gordana's lab. So basically, we want to develop a human engineered cardiac tissue model that has a macrophage, which is immune cell component in it. And because we have many human engineered cardiac tissue model before, but none of them has immune component in it. So that's our innovation. And also the other innovation is that all of our components in the cardiac tissue model is derived from induced pluripotent stem cells. So it's like more similar to an actual human tissue. Yeah, so how do we develop like the tissue model? The first step is cell differentiation. So basically we have induced pluripotent cells, and then we put different growth factors, as mentioned before in the introduction, to differentiate them into cardiomyocytes, fibroblasts, and macrophages. And then we put them into a scaffold. We use, in cardiac tissue, we use hydrogel as scaffold. And then we basically combine all of the cells together and let them grow into a tissue. And then after this, because our tissue is derived from stem cells, they are more immature, which means that they are more to the fetus status, which means that they don't beat, because in uterus, your heart doesn't beat. So we use an electrical stimulation to help them mature and become more to the adult heart tissue. Oh, and the culture and simulation, the maturation takes 28 days. And after 28 days, we did an immunostaining to see if there are still macrophages resident in the tissue. And next, I want to talk about the bioreactors or the platforms we use, which is Millipillar. This is a platform we specially designed for cardiac tissue in our lab. So basically, on each platform, there's 6 wells in it, and in each well, there are 2 pillars. And then we want the tissues to form around the pillars so that in later force measurement, we can measure how much the pillars deflect, so that it can reflect how much force the cardiac tissue can provide. Yeah. So because we need to stimulate the tissues to help them mature by putting electricity on them. So we run through 2 carbon rods, which are the 2 carbon rods, into the platforms. And then we also punch holes on each end. We can see here, this is an actual picture of the platform. We punch holes onto the 2 ends, and then we connect the platinum wire onto each end. And then we put the electrical wires, like we clip the electrical wires onto the platinum wires so that we can give them the electrical stimulation to help them mature. Okay, so in this paper, to confirm that there's still macrophage presence within the cardiac tissue after 4 weeks, they did some RNA sequencing to make sure that the pathways or the genes that are specifically expressed by macrophages are still there. So yeah, this is like, people should be more familiar of this than me. So yep. And then after that, we put the platforms under the microscope, and then we took videos of them. So in the first 30 seconds of the video, we don't put stimulation onto it, and on the second half of the 1 minute, which is the second 30 seconds, we put stimulation onto it to make all the tissues beat at the same rate. And then we take videos of them, and then we have a specially designed program to see if to measure, like the program can capture how much the pillars deflect from their original position to measure how much the cardiac tissue provide force. So the program measure the force and then gives us all the results. So we can see that we get results for mean active force, mean active stress, mean force, and mean stress. And there are a lot more. These are just the more significant results. Yeah, so we can see that in all the with macrophage groups, the tissues can provide more force, which means that they have better performance. And then to figure out like what's the molecular details that makes the tissues with macrophage perform better. They did the this is the KEGG, I believe, pathway, and then they figure out that it is actually the beta adrenergic pathways that are altered, so the tissues have better performance. And then to confirm that is the beta adrenergic response that's that better like modulate the cardiac tissue. They use isoproterenol, which is a kind of adrenergic agonist that can enhance local calcium ion release in cardiomyocytes to like strengthen the signal within the tissue so that they can see how much calcium level are in each group of tissues. So that they found out eventually that the presence of macrophage leads to a shorter calcium transient decay compared to the without macrophage controls. So the next step, which is the thing I'm doing now, is that now we have the model that has a immune component in it. So we want to put the model into an actual disease setting. So the kind of injury setting we choose is the ischemia reperfusion injury, which is the injury heart tissue get when it suffers from a heart attack, which basically means that for some time there's no blood flow into it, and then to actually rescue the situation, we need to pump blood and oxygen back to it. So that process actually gives the tissue damage. So why is it? It's basically because like the microenvironment of the tissue changed drastically in a very short period of time. It's very similar to when you freeze down some cells and then thaw them. There will be some cells that like get stressed or shocked. So it's basically the same. So I want to see if the addition of macrophage can better help tissue recovery after this ischemia reperfusion injury. So how we do this is that we put both groups of tissue into the ischemia chamber, and then we flush the chamber with nitrogen to make sure that the oxygen level in the chamber is low enough. And then we cap the tissues in the chamber for 24 hours, and then we take it out and cap in the normal chamber for 24 hours. And before we measure the force before we put in the ischemia chamber, after we put in the ischemia chamber, and after they are in the normal, which is like with oxygen contacts for 24 hours. So, and then we compare the 3 force measurement, and also we do some live death staining to see if there are like many macrophage that dies in the tissue. And then we do analysis to all the data, and then we also keep the tissue for another month to see how the prolonged effect of macrophage is. Yep, and that is basically my research, and then also the research our lab is focused on. Thank you.
That's very cool. 我想问一个问题，就是 so like a big problem, not like a big challenge in like iPSC generated tissue, is like 就是他们 generate the cardiac tissue, 就是 cardiomyocytes 没有办法 be in sync. Are you guys trying to like tackle this right now? Just curious.
Wait, what do you mean that they can't be in sync?
Beat in sync 就它没有办法同时跳动。
哦，OK，
Yeah，
就是你的意思是说，它这个 tissue 里面的所有 cardiomyocytes 不能够同时跳动吗？
对对对，就 like an actual heart， 它所有的细胞是 beating in sync 的，对吧？但是就是generate 出来，就是以前有一些 cardiac tissue regeneration paper 是，但是现在就是还是没有办法达到，就是说所有这些 cardiomyocytes 都 beat in sync。So I was just wondering 你们 lab 有没有就是？
其实我猜测，因为它这个 differentiation 呢，它 理论上它因为心肌细胞它是有很多种，它有 ventricular and then something else。但是呢，我们的 differentiation 呢是 mainly focus on 就是我们定向的把这个细胞直接分化成了 ventricular。我觉得应该是不同的，如果它differentiate 出来的是两种不同的 cardiac tissue 的话，因为可能你的心脏有收缩，然后会有几个 step，所以它会就是 doesn't beat in sync。所以我觉得如果你特意的把所有的cardiomyocytes 都分化成同一种，比如说我们 focus on 就是 ventricular cardiomyocytes 的话，它可能会稍微好一点。但是确实是因为这样子的话，你不能够完全 mimic actual heart situation，因为你不可能心脏里面只有一种 cardiac myocytes。I think that's the first step, but no, we're not tackling that like differentiation aspect thing, I guess.
Okay, interesting. See. This is very interesting paper.
All right, I will go next. Share some pictures. Okay, is everyone able to see my presentation?
Yes.
All right, so thanks everyone for coming. I'm just gonna really quick go through a few industry things for tissue engineering. So the tissue engineering industry, I'm just gonna go through an overview for now. So since the term tissue engineering was first introduced in 1993, this field, along with regenerative medicine and cell therapy, has evolved from early stage concepts to widely used clinical applications. Tissue engineering broadly combines engineering and biology to restore or improve tissue function, including the development of artificial organs and biomaterials, as well as cell therapies. The growth in this field is evident from the substantial number of research publications. There were over 1.2 million results for tissue engineering alone on Google Scholar, so that's a lot of papers. This underscores the field's rapid expansion for over the past 25 years. Tissue engineering is inherently interdisciplinary, drawing from a variety of scientific and engineering disciplines to achieve the complex goal of regenerating organs and tissues. In my review, I will be focusing on the analysis of public companies in the US due to the challenges in accessing financial data from international companies and private ones. So it's mostly just going to be like big companies. And my goal is to provide an updated view on the current state of the TE industry, highlighting its continued growth and maturation as it stabilizes within. To get started with some industry-wide data, the sales distribution across tissue engineering product types have been segmented into three main segments. Companies that use a predominantly one material, including proteins and growth factors for tissue engineering applications, were classified as a biomaterials company. Companies that incorporate cells, so whether they are autologous, allogenic, or xenogenic, into a biomaterial for tissue engineering applications, were classified as a cells and biomaterials company. And for those companies that use altered or unaltered stem cells alone, the classification of stem cells. And so we can see from the graphs that figure 2 presents the sales distribution across different TE product types, with biomaterials-based companies clearly dominating the market, which really means that they are using proteins and growth factors to induce the growth of certain cell types on a artificial construct. And this trend highlights the maturity and established demand for biomaterials in the industry versus cells and biomaterials, which really means that you're trying to generate cells from scratch. Stem cells, as the name suggests, these industries are still emerging with less market impact so far. Over here, we're going to look at figures 3B and C, which provide insights into the industry composition and employment. Despite the higher number of stem cell companies, biomaterials-based companies employ the largest workforce, indicating that biomaterials are not only leading in sales, but also serve as the economic backbone of the tissue engineering industry. When it comes to research and development investment, figure 3A reveals that stem cell companies are heavily investing in research, even though this hasn't yet translated into major sales. This suggests that the stem cell companies are focused on future innovation, which could lead to significant breakthroughs, albeit with higher risk. And lastly, service companies, especially those in stem cell services like cord banking, have a substantial financial presence driven largely by key players like Celgene, which really are companies that provide a service where they bank cells of yours and my meetings. Where they bank your cells and you can probably manipulate them later on. And also this would also include the companies that provide the cryo freezing services after you die. This sector, although concentrated, is crucial to the industry's overall financial health and may signal areas of future growth or consolidation. Okay, my meeting is about to end. I'll try to get through this as much as possible, and then we can get back together for questions if that is needed. Okay, so for sales and the distribution of operating costs, we can see a clear trend in sales distribution across the different developmental stages. Biomaterials are dominant in the commercial stage with 99% of sales, so 99% of revenue and deals generated are within the sector. Biomaterials are dominant, but we can also see stem cells taking the lead in both clinical and preclinical stages, contributing to 77% and 74% of sales respectively, which points to their potential and growing focus of new therapy development, which really just says that stem cells is like the upcoming field. Figure 4D, shows that more companies are at the commercial stage with a strong emphasis on biomaterials. However, the significant number of companies in clinical and preclinical stages, particularly those focused on stem cells, reflect the industry's active investment in developing future therapies. In terms of operating costs, the commercial stage sees the highest expenses, largely due to the predominance of biomaterials-based companies. Meanwhile, stem cell companies are driving the spending in clinical and preclinical stages, highlighting their commitment to advancing through rigorous development and trials. And lastly, while the number of interventional clinical trials in the US is still relatively small compared to the total, the presence of tissue engineering related studies marks the field as an emergent area with the broader context of medical research. This suggests that the growing interest and potential for innovation in tissue engineering and regeneration. Next up, I'm just going to go through a few companies of significance, a few that I found personally to be very intriguing to talk about. This first one is called BioTissue, and they have a few different products. I will be focusing on Neox and Clarix. So BioTissue was originally founded as Tissue Tech in 1997 and rebranded in 2022. It's based in Miami, and it's a biotech company focused on regenerative medicine. They specialize in using human birth tissue to develop products for ocular surface disease, which really means anything that irreversibly damages your cornea, and any unregenerated tissue in your eyes, surgical applications, and wound. So their key products include Procara for dry eyes disease, an Aminograft for ocular surgeries, and Neox for treating complex wounds. A major differentiator for this company is Cryotech, which is a cryopreservation technology and preserves the HCHA-PTX3, it's an alphabet soup, complex that is crucial for healing, ensuring their products maintain high structural and functional integrity. So this company has shown clinical success in improving outcomes in eye surgeries and wound treatment, supported by sustainable practices like the Sharing Miracles birth tissue donation program. And this combination of innovative technology, proven efficiency, and sustainability drives to market success. I want to just zoom in on the Cryotech preservation tech real quick. It is designed to preserve the structural and functional integrity of human birth tissues. So specifically the extracellular matrix components, of the amniotic membrane. So this includes critical molecules like the heavy chain hyaluronic acid, pentraxin 3, which is the alphabet soup that I mentioned just now, and also includes collagens, fibronectin, laminin, proteoglycans, and various growth factors. Molecularly, cryotech works by preventing the degradation of these key ECM components during the preservation process, which is crucial for maintaining the tissue's natural healing properties. Unlike traditional heat dehydration methods, cryotech ensures that the amniotic tissue remains in its native structure and biological activity, which is essential for regenerative healing. This process is particularly effective in promoting wound healing, reducing inflammation, and minimizing scarring in medical applications, such as ocular surgeries, where you really don't want to have a scar because that would screw your vision, right? The preservation of the HCHA-PTX3 pathway is especially important because this complex plays a significant role in mimicking the fetal healing, which is known for its anti-inflammatory and anti-scarring properties. This makes cryotech preserved tissues highly effective in therapeutic applications, proving better for patient outcomes compared to other preservation methods. Next company I'm going to talk about is Cellular Logistics. Cellular Logistics is an innovative biotech company based in Madison, Wisconsin, specializing in the development of advanced biomaterials for cardiac care. And so, ding, 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 this actually connects very nice to what Wei was talking about, right? Because their flagship product is actually called CFX, or cardiac fibroblast matrix, and is literally designed to address the need for a regenerative healing solution after having a heart attack. So CFX is a therapeutic biomaterial designed to prevent heart failure following a myocardial infarction or a cardiac attack. The product leverages induced pluripotent stem cell or iPSC technology to create a cell-free matrix that supports the heart's healing process by preventing pathological changes and facilitating tissue repair. So I'm not sure, like, I tried to search for a relevant literature, but very little was shown on the molecular mechanisms of CFX. But more or less, I think the whole idea behind it is preventing inflammation in the heart and making sure that the inflammatory molecules circulating the heart while it's in its healing process doesn't damage the heart further, right? So CFX works by mimicking the natural ECM matrix of the heart, which is crucial for maintaining tissue structure and function after a heart attack. When applied locally to the damaged heart tissue, CFX integrates with the existing tissue and promotes cell retention, reducing scar formation and aiding in the remuscularization of the heart. So one thing that happens after your heart failure is your heart basically goes into lockdown and all the inflammatory molecules try to come in and fix the tissue by generating scar tissue. And that is absolutely what you don't want for your heart because it has to be the original the original muscle tissue that keeps the heart beating. And so you have an excessive amount of the scar tissue, your heart function is going to be significantly impaired. So this is what CFX does. It prevents those inflammatory molecules from damaging the heart further and attempts to regenerate the muscular tissue, right? So this is particularly important in chronic heart failure because the damaged myocardium can be restored to improve heart function. One of the key features of CFX is its immune privileged nature because it is literally generated from your own cells, making from your own stem cells, which makes it a safer and more accessible option for patients because you would not need immunosuppressive drugs to deal with implanting foreign cells or anything like that. It's also really versatile because it could be delivered as an injectable particulate or patches, right? With customizable sizes tailored to fit specific clinical needs, so it can address a large range of heart damage problems. And so yeah, that's CFX. And this company, Cellular Logistics, also has a partnership with Allele Biotechnology, and they developed something called Solus, which is a new regenerative product focused on treating ischemic heart disease. I'm actually highly suspecting that the Gordana lab could have something to do with this company because it looks really similar. I don't know, just a speculation. Okay, so
sorry, sorry.
yeah, no, the meeting is about to end, so I'll just stop the meeting and then come back.
呀，我就想问他这个cardiac fibroblast matrix 它这个 iPSC technology 用的是 patient derived iPSC 还是就是
Yes, it's custom made, so case by
OK
case. Yeah.
OK interesting. That's very expensive.
Yeah, I know.
Yeah.
Most of these companies, like if you actually look at their websites, it's pretty shady. And I'm thinking like it's probably like one PI who spun out like his life's work in the lab and made this company
Mhm.
that's going to make absolute bank because you know your product is mainly facing like probably billionaires or millionaires who really want to live a little bit longer. So that they'll spend like a whole lot of money to get these like really new cutting edge treatments. Um,
Interesting,
yeah,
interesting. Okay.
I think that's generally how they're money making. Um, anyway, so next up, Vericel Corp. This one is a little bit different. It's also really interesting. So Vericel is headquartered in Cambridge, Massachusetts, right next to Harvard and MIT. It's at the forefront of advanced therapies for sports medicine and severe burn care. So it was originally founded as Astrom Biosciences in 1989, and the company transformed after acquiring Sanofi's cell therapy and regenerative medicine business in 2014, and has since been rebranded as Vericel. They also have a few different product lines. So I'm just going to mention MACI, which is an innovative treatment for knee cartilage repair utilizing a patient's own cultured chondrocytes on a porcine collagen membrane, which really means you're taking the patient's own cells, you're cultivating it ex vivo, and then once that construct grows, it will be tailored to a certain shape. Again, custom made, very expensive. And then it will be custom fitted to cover the damaged area for optimal joint healing. Epicel provides a life-saving solution for severe burn victims by expanding a small sample of the patient's skin to cover extensive burn areas. And Nexobrid, derived from pineapple enzymes, offer a non-surgical option for selective removal of eschar in severe burns, preserving healthy tissue and accelerating recovery. So yeah, I guess what's interesting about this company is that like their Epicel and Nexobrid technologies, when combined together, have a very high efficiency for saving lives in burning victims. So as a little bit of context, burning wounds are extremely hard to treat because the area of damage is usually really wide, and then it burns down your barrier between like your inner tissue or large area between the outside and your tissue. So inflammation and infections are very likely, and also it's really hard to treat burns via surgery to remove the damaged skin because a the penetration of damage is very varied. So that means you can have one like small area where nothing is damaged at all, and then another area where basically your subdermal fat can be burned away. So it's like so you can never very precisely remove like the right amount of damaged tissue away from the skin, if that makes sense. So what Nexobrid does is it's an enzyme that is somehow able to identify dead cells, right? It's able to identify tissue that has been damaged by burn. And then from those enzymes, they are able to very selectively remove the damaged tissue, which opens up a lot more possibilities for burn recovery and also minimizes the chances of infection because you can remove all the damaged tissues ASAP and do it at a more effective rate as well. So yeah, and then I think Epicel is more so just like your run-of-the-mill like normal tissue engineering concept where you take a small sample of the cells, you grow it up in like a petri dish essentially, and then you use that grown up sample the um to cover up the burned up area so that the cells can relocate and start growing again over the damaged areas as such. So what makes Vericel unique, right? It's very focused on patient-specific autologous treatments, so own body treatment that enhance biocompatibility and minimize the risk of rejection. also at the cutting edge of the regulatory process. This company is insanely good at getting FDA approvals. They have anticipated FDA approvals for Macy, Nexobrid, and then their Episode technology is already approved. And so yeah, they have a lot of potential for dominating the burn treatment market segment. And so yeah, a few takeaways that I want to mention. So tissue engineering and the future of tissue engineering based therapeutics, right? In reviewing the trends and developments of tissue engineering, it is clear that while earlier reviews provided valuable insights, they lacked reproducibility. And this is talking about the paper. Our approach has focused on the transparency of public data, and that has also limited direct comparisons that we can make, even though they all have their patent forms and whatnot. But based on the fact that they work in very different industries, it's hard to make direct comparisons. However, there are a few key trends that I want to mention. So stem cell companies are still leading in research and development spending during the early developmental stages, while most biomaterials companies focused on autologous development have already entered the commercial phase. So they are running at higher operation costs. Interestingly, even though tissue engineering is a small subset of clinical trials in the US, there's a significant industry sponsorship at 77%. And so this is an industry that is really heavily connected to the academic world, highlighting a strong commitment to advancing TE products to the market. And it's also one of the industries that is most heavily capitalized, if that makes sense, from your academic labs. However, external factors like FDA regulations and CMS reimbursement policies heavily influence TE product sales. For instance, products like Dermagraft and Apiligraph were negatively impacted by new CMS guidelines, which placed them in less favorable reimbursement categories, discouraging their use despite their advanced technology. We're not exactly sure why that happens. Why the FDA and the sort of insurance industry has really worked against tissue engineering based therapies. But it's still important to note that the number of clinical trials doesn't necessarily predict the future market success of the TE industry. Continued analysis over time will be crucial in understanding why and how many TE products successfully reach the market and generate a heavy amount of revenue, with regulatory and reimbursement policies playing a critical role in their future success. So with that, thank you so much for listening. That has been me and the tissue engineering industry. 如果有问题的话，现在可以随时问，然后问武艺和 Cassie 的话，也都可以再补问啊，各位。没有问题吗？大家？
谢谢大家，谢谢大家。你要不要先关掉 recording，然后？